Hello and welcome to the second major discussion of molecular biology issues, right? Last time we talked about DNA replication. That is a completely different process relative to what we're talking about today. I mean, all you need to do is think about the whole importance of the process. DNA replication is completely important because we need to copy DNA. Okay, we can't undergo cell division unless we've got 2 copies of the genome, because every cell needs to have a complete 6 billion base pair copy of the entire human genome. Okay, and making 6 billion base pairs is not an easy task. It's a whole process that has to occur with very little mistakes, with very few mistakes, should I say. And it's going to happen. In particular, I mean, if you think about the timing, it's another place you can realize that this is a completely different process from transcription. This is a process that's happening during the S portion of interphase, in which at which time the nucleus is pretty much got to give its entire all of its resources has to go to this process of DNA replication. In other words, it's kind of its own little empire. During the S portion of interphase, DNA replication begins. We open up those replication loops. They spread out pretty much. We get continuous and discontinuous DNA synthesis, and then we end up at the end of the process with 2 identical copies of the double-stranded molecule that's 6 billion base pairs long, right? Crazy. And that's happening in a relatively short amount of time during this S portion of interphase, right? And so you keep in mind, I'm going to change colors entirely to make you realize that what's going on with this lecture is something that's happening relating to a different process. In fact, it's going to revolve around the central dogma. The central dogma is like one of those buzz terms that is tossed around by molecular geneticists. Anybody who's taken a biology class knows that central dogma is supposed to describe the flow of information through the cell, where we have information that starts off in the DNA. DNA information is transcribed to make a molecule of RNA, which is a carrier molecule. It carries the genetic information. In other words, we have the master copy of the information in the form of DNA. We'll make a copy of the information that we need in the form of RNA, and then we use the code of RNA, the sequence of bases in the RNA, to make a protein, which is the final gene product. Okay. And so this flow of information from DNA to RNA to protein is what's referred to as the central dogma. And if you're clever, you could probably describe that. You could probably come up with a very brief version of that to basically give the whole picture of molecular biology and gene expression in 10 words or less. Okay, can you do that? Yeah, well, maybe not right now, but perhaps by the end of our module 14, you'll be able to do it pretty well. Okay. So the other thing about this, this is not completely addressed in the textbook, but when in the cell cycle do we see the majority of transcription, right? This is basically gene expression. That's not how you spell gene. Okay, and so at what time during the cell cycle do we have the majority of gene expression? Well, it's not during the S portion of interphase, because we know that during the S portion of interphase, the entire genome is busy being copied, right? It's not during mitosis, because during mitosis, the nuclear material is in the process of getting packed up really tightly. It's like we're taking the entire 6 billion bases worth of DNA, packing it into really tight little suitcases, and then having those suitcases magically split apart into two identical suitcases that are going to go off into the daughter cells and then unpack, right? And so we're not going to have a whole lot of gene activity, at least not at the level of DNA being read, either during the S portion of interphase nor during mitosis, which basically leaves us with the only other two portions of the cell cycle, which would be G1 and G2. Okay? Both are associated with growth. G stands for gap, but both are associated with growth. And I guess I should also put in G naught. Oops, and do that. Which corresponds to those cells that are not allowed to continue with the cell cycle. They basically never go into S, and so they're pretty much arrested in kind of like a late G1 phase, right? But it's these cells that are actually doing the things the cells need to do. They're expressing the genes, they're making the proteins, they're doing everything that needs to happen, okay? And so before I actually show you the some pictures from the textbook, you might also consider where in the cell does transcription occur. And well, it has to happen where the DNA is, and the DNA is located in ... Well, yeah, I mean, if you're thinking about a eukaryotic cell, it will be the nucleus. And prokaryotic cells don't have membrane-bound nuclei, but they do have a nucleoid region where all the DNA is packed, and that's going to be, you know, wherever the DNA is going to be where transcription occurs. Okay. And so now would be a good time for us to switch over to our textbook. And we're looking at figure 10.6a. This is in chapter 10, part 6. And this is basically showing you the flow of information in the cell as described by Archibald Cahoud, who is an English physician. It says right here at the bottom of the page. But generally, the idea is that we've got the DNA, the information code of the DNA, giving rise to a molecule of RNA. That's the transcription part. And then yet later on, after the RNA transcript, after this piece of RNA is converted into a full-on messenger RNA molecule, at that point, it's going to be exported into the cytoplasm, the cytosol, where it's going to be taking part in translation. And that's going to happen at the ribosomes. We kind of briefly talked about how that works early when we were discussing cell biology. Okay, and so basically 10.6, 10.7, 10.8 are kind of like descriptors, just kind of giving you a little bit of a pep talk about how information is going to be flowing through that process. And it kind of gets us into the process of translation. We'll be talking about translation next. So yeah, it's a really good idea to read through these sections, because I'm not going to ask you to come back and reread these sections when we actually talk about the process of translation, right? They do their genetic code in this circular chart. Traditionally, the code is in a rectangular chart, and that's going to be the one that I'll be talking about when we get into our live session. This round chart is great, but it just, well, it just is, requires a different skill set or protocol to follow through. And so this is basically another picture of how we're going through the processes of transcription and translation. The DNA is this blue molecule at the top. During the process of transcription, an RNA molecule, which is the thing that's shown in red, is produced. That's happening in the nucleus. And then after the messenger RNA molecule goes out into the cytoplasm, the information carried in the RNA molecule is going to be translated into a sequence of amino acids. Okay, what we're looking at here is AUG is going to be translated into the amino acid methionine. AAG is going to be translated into the amino acid lysine. UUU phenylalanine. Right? And so these little purple shapes at the bottom that are linked up with peptide bonds. This is our polypeptide that's in the process of being formed, right? And so you get these schematic pictures, many different perspectives on the processes of transcription and translation. We get some cute little cat pictures at the very end. And then we get to 10.9, which is the actual nuts and bolts descriptor of how translation is taking place. And basically short version here, I recommend that you look through this on your own in great detail. But yeah, we've got RNA polymerase. RNA polymerase is, well, if you break that down, what are we doing? We're polymerizing and we're making a molecule of RNA, right? So this basically tells you that we make RNA, the polymer, from mononucleotides, from ribonucleotides, right? And so the polymerase is going to be starting the process on a promoter. This is promoter, important thing. This is where it starts, where transcription starts. Okay. And then once we have the RNA polymerase landing on the promoter, which the promoter is part of the DNA code, it starts to move forward. It's going to land here and then move in this direction. And we'll have the process of elongation. Right? Elongation is the second part where we're actually making a molecule of RNA. That's the pink molecule RNA down here. RNA is being copied using complementary base pairing, right? See that here, complementary base pairing. Okay. And the RNA molecule is being shot out as the RNA polymerase is, pushing its way down the DNA molecule, right? So elongation refers to the fact that during this process, the newly made RNA is being elongated. It grows longer and longer and longer. It starts off being relatively short, and then as the RNA polymerase moves down towards the terminator portion, which we're getting at, which are basically moving towards the terminator, the RNA molecule is getting longer and longer, hence the name elongation, right? And then when we reach the terminator sequence in the RNA, the RNA polymerase falls off and the RNA is released, right? And so it's actually a pretty basic process. I'll kind of walk you through this again, because a little bit of repetition is a good idea. The process of transcription is very, roughly similar to the process of DNA replication, except we're making a molecule of RNA instead of a molecule of DNA, and we're not doing the whole genome. We're only doing the gene, the transcription unit in the genome. So as opposed to being like 6 billion base pairs being copied, we're making an RNA copy of maybe a few thousand base pairs, right? And then we're releasing that, and then we can make, and after that's done, realize that this is something that happens over and over and over again. When this gene needs to be expressed, this is basically the gene that's being expressed. If that gene is turned on, the promoter will be attracting more RNA polymerase. So the RNA polymerase will be landing, will be making many copies of the RNA, which is then going to undergo RNA editing and eventually make it out of the nucleus, right? To then engage in translation and basically to make the gene product, the proteins. All right. So let's go back to my whiteboard share. And here we are again. Make this a little bit wider. Okay. What is the thing that triggers translation? Okay, this is a really good question, and we're going to save that until module 11? Okay, it's such an important question. I know that it's something that's bugging you right now. You're dying to know about how we actually initiate some genes, but not others. But you have to realize that that's kind of like, the whole answer. That's the holy grail question, right? If you understand that, then you understand the way that biology works at the molecular level, right? And so it's such an important question. We're going to dedicate pretty much an entire, let's say module 11, and then module 15, right? Yeah, the next module after the one we're in right now, right? We've talked about what happens in elongation, termination, and initiation, elongation, termination. And then the next thing that's going to concern us potentially, and not a huge point, is what happens next. So let's go to a different whiteboard. And I want to draw for you, using the same color coding that was there in the textbook, molecule of DNA, hoping that if I draw a molecule of DNA, you'll follow suit, do the same thing in your notes. Okay, so this is a molecule of DNA. It's going to keep on going in both directions. I'm only drawing the portion that's of interest to you. And maybe this might be the promoter region over here. This might be the termination region over here. I'm not going to color code them differently. Okay. We know that the RNA polymerase was kind of drawn like a little bullet like this, a little groove here. RNA polymerase okay. It's going to land on the promoter region, and basically we're going to be opening up a little blip so that will expose the DNA on one side of the molecule. Okay? And so now I'm going to draw this again, go back to my DNA picture. And so this might be the promoter region here. But I'm going to draw it a little bit open so you can see what's going on. So the blip is opened up, and we're basically exposing the bases here, right? So we'll have maybe a T over here, which basically means we'll have an A over here. We'll have a G on one side, we'll have a C on the other side. Remember the rules of complementary base pairing. C over here, G is going to be over here, A, T. So maybe it's going to be C, C, T, A, A. So this will be G, G, A, T, T, and we're continuing to unwind, right? The transcription is happening from left to right here, and so we're going to be making a new molecule of RNA based on the exposed base that we have on this strand. Okay? So this strand is going to be the one that's red. Strand A, which is red. Okay. Strand B which is basically just, it's not red, it's just occupying the space opposite of the strand that's red. Right? This would be unread. Okay. And we actually have names for this. We call the strand A, we call this the sense strand. And strand B, we call it the anti-sense strand because it's opposite of the sense strand. Right? That makes sense to you? I mean, it should, okay. And so what the RNA polymerase is doing is just coming in and it is hooking up the bases, right? Opposite of a T, you'll have an A. Opposite of a G, you'll have a C. Opposite of a C, you'll have a G. Opposite of an A, you'll have a U, remember? Because we're talking about RNA. And opposite of C, so you can basically visualize these mononucleotides, these ribonucleotides coming in, and it's going to be continually forming in this direction from left to right. And so this is going to have to be the 5' end of the RNA transcript. It's going to be the 3' end, right? And what the RNA polymerase does, what this molecule here does, is it comes and zips all these guys together to create a single, a united molecule of RNA. And then that molecule is going to be released, and we're going to be closing up the DNA on this side. And so if we come back like a microsecond later, the DNA will have wound up a little bit. There it is. We still have bases out here that are being exposed in the DNA. This is still the sense strand. The antisense strand is over here. We're still putting molecules of RNA together, right? We still have the RNA polymerase doing that, but the RNA molecule that is produced is coming off the side. So this would be the A, C, G, U, G. This would be the 5' end of the RNA molecule that's being formed. RNA polymerase, our busy little enzyme, is going to be over here, okay, putting the RNA mononucleotides together, and eventually we're going to be getting to the terminator sequence. The termination portion of the DNA is going to be there, and at that point, the whole thing can close up, and we'll be able to we'll release the raw RNA transcript, 5' one side, 3' on the other. Call this the RNA transcript. It's not a messenger RNA molecule yet. It might not even become a messenger RNA molecule. But remind yourself of what transcription means, right? It means we're copying something. We're basically taking information in this book and writing an exact copy of the same exact sequence of characters. And basically that's what's happening during the process of transcription. Okay. So again, I mean, it's an important enough process for you to realize the questions that we still need to address. Well, why does this happen? How do we initiate? What triggers? Remember I said that we promised we have to talk about the trigger for gene expression, which is an important consideration because not all genes are being transcribed and expressed. Any single cell in your body is expressing actively only a tiny percentage of all the genes that are there in the genome, right? That's how you end up with skin cells doing what skin cells do, and bone cells doing what bone cells do, and nerve cells doing what nerve cells do. I mean, the only way we can get that differentiation of function is by having different genes expressed. And so the mechanism that biological organisms like you use to trigger the appropriate gene expression in the appropriate cells, that's a big question, right? Well, all we're saying here is that if we do trigger the expression of the gene, it's going to transpire in this particular way, right? Now, once you get the RNA transcript, okay, this could do a couple of things. If it's going to become messenger RNA molecule, okay, if it's going to carry a message that's going to ultimately give rise to a protein gene product, it has to undergo RNA editing. Okay, in the nucleus, okay, before it eventually becomes an mRNA. Okay. This is what's described in Chapter 10, Section 10. We'll be talking about that particular process in greater detail in the next module. I want you to read through it so you understand what it's all about, right? Okay. Now, the RNA transcript might not necessarily be producing a protein gene product. Maybe the gene product might be either tRNA, which is one of the pieces, which is actually many pieces that are required for the process of translation, or it might actually become part of the ribosome itself. There are parts of the ribosome that are made up of RNA, and so we've got a really large amount of RNA molecules that are responsible for the production of ribosomes. Remember where that happens in the cell? Starts with an N, rhymes with nucleolus. Okay. That little dark spot inside of a nucleus that we have in stained cells. Yeah, those are the portions of the DNA where you have a lot of gene activity because the transcripts from those cells, from those genes, are going to become ribosomal pieces, either our ribosomal RNA, but also we have ribosomal proteins that are also transcribed in those same areas, right? And so chapter 10, section 11, chapter 10, section 12 kind of gives you a little bit of a taster of what else besides a message carrying RNA is being produced in during the process of transcription. And so these are the answers. And not every RNA transcript is going to ultimately become an piece of information for a protein. Okay. Now the last little bit here is that if this happens in a prokaryote, right? If we're talking about DNA in an E. coli cell. Remember, E. coli is a prokaryote, meaning that there's no nuclear envelope separating the place where transcription is occurring from the cytoplasm where the translation is taking place. So in other words, transcription and translation are taking place in the same location, right, in the same spot. And so we don't really have any opportunity for DNA editing, right? We can't say, okay, well, ribosomes, you have to wait for me to get the RNA molecule completely ready. That's not a possibility. And so with E. coli, you've got this really cool little thing where you can actually see the DNA. You've got a good enough microscope, you can see this. So this might be the DNA. Okay. And then the RNA polymerase, this might be the promoter end over here. So the RNA polymerases might be doing their thing, and they keep on landing here. RNA polymerases keep on landing at the promoter region. They move down. They basically chug along this direction, right? You'll have this procession of RNA polymerases. RNA pol is the way that molecular geneticists refer to it. And as this is going along, the RNA molecule gets longer and longer. So this might be a relatively short RNA molecule. Over here, the molecule might be a little bit longer. Over here, the molecule of RNA is really long. And over here, the molecule of RNA is being produced is it's almost done, right? And while that's happening, we actually have a third set of things going on. Let's pick a new color altogether. Let's make it green, right? And so ribosomes, okay, which is where translation takes place. Okay. Ribosomes can land on the 5' end and start making protein. Okay, so here's a ribosome that has just landed at this end. It starts to make a protein. By the time it gets over here, the protein is much longer. By the time a ribosome gets over here, the protein is really long. And so you get these structures in electron micrographs that look like little lamp brushes. Okay, continue this drawing. And so if you're following along with this here, you're actually seeing both the processes of transcription and translation taking place. This is it's a little bit more than just trivia. What I'm giving you is an opportunity to kind of visualize who the players are in the processes of transcription and translation, right? The ribosomes are doing the translation part. This is, it's basically reading RNA molecules, RNA information, giving rise to protein. Right? In this diagram, RNA polymerase is responsible for transcription. We're taking the information in DNA and making a molecule of RNA in the process, right? And so, while this is a really nice picture, it's only something that we can visualize because bacterial cells don't have a separation between the locations of transcription and translation, because there's no nuclear membrane that separates the process of transcription from the process of translation. So in contrast to eukaryotes, where this whole process is going to be taking place in the nucleus, and then after we finish our messenger RNA molecule, or after we finish making our tRNA molecules or RNA molecules, those molecules are going to be exported outside of the nucleus, and then they can undergo the process of translation. Okay, this is a good place for us to stop. We'll be picking up the story with a more detailed picture of translation in the very next video.